Good afternoon, friends. Today we will discuss one of the very important staining technique in microbiology. It is called simple staining. As we know, bacteria are colorless. So, in order to observe these bacteria under the microscope, we have to stain them. If we want to determine their morphology, if you want to determine their arrangement, we need to color them. And this coloring procedure is called a simple staining. And there are different methods of staining in microbiology, out of which we will discuss one of the method, one of the technique. It is called a simple staining. So let us discuss in detail what the simple staining technique is. In staining technique, whatever may be the type of staining technique, certain colors are used and they are called a stain. So, a simple stain is used in simple staining. A single type of stain is used in simple staining. This stain is usually an aqueous or alcoholic solution of a basic dye. The important thing is the stain used in simple staining technique is basic in nature. It means it possesses a positive charge and the charge on bacterial cell wall is negative. So, there will be an attraction between this negatively charged bacterial cell wall and a basic dye which is having a positive charge. So, whatever type of stain that is used in simple staining technique should possess a positive charge. The most commonly used basic stains used in simple staining, they are methylene blue, crystal violet, carbolfuchsin. All they are basic stains. So, ultimately they will bind to negatively charged bacterial cell wall because they are having a positive charge. This simple staining technique is very easy to perform and it highlights the entire microorganisms so that the shape, size and arrangement of the bacterial cell is visible. And in simple staining, one stain is used and all types of bacteria appear as the color of that stain when viewed under microscope. This is very important concept in simple staining. A single stain is used in simple staining and color of this stain will be the color of bacterial cell. Suppose a blue colored stain is used. At the end of staining technique you will find the color of bacterial cell will be blue. If a red colored stain is used, the color of the bacterial cell will be red because a single stain is used. And this staining technique, the importance of the staining technique is that it will help to observe entire microbial cell shape, size as well as arrangement of the cell. Principle of simple staining. So, when basic dyes are used such as methylene blue, crystal violet, they will bind to negatively charged cell wall. Basic dyes are having positive charge like methylene blue, crystal violet. So, when they are used, they are applied on the surface of bacterial cell. These positive charge on this basic stain will bind to negatively charged bacterial cell wall and the bacterial cell will get the color of this stain. There will be an attraction between positive charge on bacterial cell wall and a negative charge positive charge on stain and negatively charged bacterial cell wall. So, if methylene blue is used, the bacterial cells will appear blue because color of the methylene blue is blue, and crystal violet is used, the bacterial cells will appear violet because color of the crystal violet is violet. This is the simple principle behind simple staining. Procedure: take a clean glass slide, prepare a smear of the bacterial culture at the center of the slide, and then flood the slide with one of the stain. If you are using crystal violet, you can flood the plates, flood the slide with crystal violet for 20 to 60 seconds. Then gently wash this smear with tap water to remove the excess stain. This step is important, rather critical, because during this step you should hold the slide parallel to the stream of the water. Otherwise, the smear will be washed away completely. You should gently wash the slide below the tap water by keeping the direction parallel to the stream of water. That will reduce the loss of bacterial cells from the preparation. Blot dry the slide, do not wipe the slide, and then place a drop of immersion oil and observe under oil immersion objective. This is the simple procedure of the staining. At the end, the observation you can make here is if you are using crystal violet, the color of the cells will be violet. If you have used methylene blue, the color of the cells will be blue. Whatever the type of stain is used, the color of the stain will be the color of cells. Here we have used the crystal violet, so cells will appear violet in color under oil immersion objective. So this is the observation of simple staining.